Okay, so I run a bioinformatics group at the University of Edinburgh, and we've been playing around with trying to model higher order chromatin structure. And by higher order chromatin structure, in this case, what I'm talking about is high C derived structure. And so from high C interaction matrices, we typically get three things. We get interlocus contacts, of course, which I'm not paying any attention to in this talk. And as we traverse chromosomes, we also get self-interacting regions, and these sort of domains of about a megabase in size have been called TADs, that we've already heard about today. And then at a higher level, as we traverse chromosomes, we can define an eigenvector, which is a principal component of the interaction matrix and defines the overall interaction propensities of a region. And as these eigenvectors change, as they flip, as we wander along the chromosome, they also define nuclear compartments, or what you might call nuclear compartment domains. So we have two levels of domain structure here that we're interested in. Another thing that we're almost forced to be interested nowadays in is huge dumps of ChIP-seq data, and so we've been using Encode data, and there's a lot of Encode data, of course, and it tends to be tens or hundreds of base pair features arranged linearly along chromosomes. And there's a large gap in our understanding of how these locus level features at the small scale relate to the higher order structures that we see coming from high C data sets. And so what we're aiming to do here is to get some insights into how these two levels of organization interact, and in the process get a more integrated view. So here are it turns out that when you look in the best covered 3 Encode cell lines, you have 35 data sets that are perfectly matched between these cell types. And they cover histone modifications, DNAs, DNA binding proteins, and so forth. And in each of these cases, we can also find in the literature a high C data set. We then go along and process these high C data sets from scratch so that they're identically processed. And we calculate eigenvectors for each 1 megabase region around the genome. And in each of these 1 megabase regions, we also have the average signal for our 35 locus level Encode features. So we end up with a large data matrix for each cell type, describing the relationships for each 1 megabase region. And when computer scientists see this sort of data matrix, they get excited and they think about machine learning techniques. And in our case, we're talking about a technique called random forests. And this allows us to build a model here of the high C eigenvectors. So what we're asking is, how well do our 35 linear locus level features, how well do they explain the variation in eigenvectors as we wander across the genome? And this is just a quick look at the eigenvectors for some of the chromosomes. And here we've got the 3 cell types in different colors, and you can see at a glance that they're fairly well correlated around the genome. You can put numbers on this. These are Pearson correlation coefficients. And in common with just about everybody else, we find that they're well conserved, suggesting in advance that some aspects of higher order chromatin structure are rather well conserved between cell types. So here are some results of the modeling. So we've got our 3 cell types here, and what you're looking at is the empirical or observed eigenvector for every megabase in the genome plotted against the predicted eigenvector. And the predicted eigenvector is just predicted purely on the basis of the Encode locus level features. And you can see it's fairly linear. And the PCC here is a Pearson correlation coefficient of the correlation between the observed and predicted eigenvectors. And in each case, we get up to around 0.8. And so from the point of view of the machine learning community, these are models that would be said to have high predictive accuracy. And there are lots of different ways to measure the accuracy of these models, but in each case, they come out looking really very respectable, surprisingly respectable, given the frankly unrealistic assumptions that we've started off with, such as averaging the signal of all the ChIP-seq data sets across megabases. One way to compare the models is to try cross-applying them between cell types. So if we train a model in one cell type and it learns the underlying chromatin grammar of eigenvector higher order structure in that cell type, how well does it predict the higher order structure of another cell type? And the answer is pretty well. We get a decrease in the correlation between observed and predicted eigenvectors. But in each case, it declines by around 5 to 20%, and the models remain useful in the machine learning sense. So that suggests that there are broad similarities in the underlying features of eigenvector models between cell types. Another way to compare the models is to dissect out the most influential variables in each case, and that's what we have here. So we're displaying the 10 most influential, the 10 most informative variables in each model. And in spite of the broad similarities that we see in terms of cross-applying the models, we see a lot of variation in the most influential variables. So for instance, we get EGR1, a transcription factor important in differentiation and development. We get that as the top feature in the embryonic stem cell line, but we don't see it in the top 10 features for the two other cell lines. On the other hand, we see P300, which is supposed to have special roles in hematopoietic cell lines, as appearing in the top 10 features for the hematopoietic cell types, and it doesn't appear in the ES cell line. So it makes some sort of broad biological sense. But the story appears to be that in spite of the broad commonalities in terms of the underlying features, there are certain influential variables that differ unexpectedly to a high degree. Another subject that's received a lot of attention in the literature are chromatin domain boundaries. And so, for instance, in the original paper reporting the existence of TAD domains, they found a variety of features that appeared to be either enriched or depleted at boundary regions. Although these are all in different scales, you'll notice, and it's not entirely clear how significant some of these features are. So we revisited this question with our 35 matched features across cell types, and you can see at a glance that a lot of features show rather interesting patterns as you traverse a boundary. And also the 3 cell types are in different colors here. There's plenty of variation between cell types. So we devised a statistical approach to measuring the enrichment or depletion over boundary regions, and that's what we're looking at here. So on the y-axis, we've got the significance of the enrichment or depletion. Along the x-axis, we've got our 35 features, and red dots are compartment boundaries, so nuclear compartment domain boundaries, and blue dots are TAD domain boundaries. And we find the previous enrichments and depletions that were reported, but these data allow us also to look to do a meta-analysis and look across the 3 cell types and across TAD domain boundaries and compartment domain boundaries and ask what's significant across everything. And the answer is active promoter features, but also stonking enrichments for CTCF and consistent, significantly significant enrichments for this YY1. Both architectural proteins and recently shown to co-bind at highly conserved sites. So in conclusion, it seems possible to build accurate predictive models. They're broadly similar, but they differ for certain influential features, and we can find prominent features delineating domain boundaries. And this is largely the work of a talented PhD student, Ben Moore, but other people pitched in.
Thank you.